इस मॉड्यूल में हम बात करेंगे सीक्वेंसिंग की यूजिंग क्लिनोव फ्रैगमेंट एज एन एंजाइम फॉर पॉलीमराइजेशन. At the time of development of the method, or the first time when the Sanger and his colleagues described the method of DNA sequencing, only the DNA polymerase 1 was the known or available enzyme. और अगर DNA polymerase 1 का structure देखा जाए, this is basically a repair enzyme out of those three polymerases which are found in the bacteria. अब इस रिपेयर एंजाइम की जो प्रॉपर्टी है दैट इज नंबर 1 पॉलीमराइजेशन एंड नंबर 2 एक्सोन्यूक्लियस एक्टिविटी 5 to 3 डायरेक्शन में होती है. इसका बेसिक काम जो है वो ड्यूरिंग और आफ्टर रेप्लिकेशन जो ओकाज़ाकी फ्रैगमेंट्स हैं उनको लिंक अप करने के दौरान प्राइमर की रिमूवल और गैप फिलिंग इज अ फंक्शन ऑफ पॉलीमरेज 1. नाउ इफ वी look at the structure of the primer and we remove one of the component which is responsible for exonuclease activity and only the component left behind having the polymerization ability, that is known as Klinov fragment. This Klinov fragment was a good and only choice at the start of the sequencing methods when Sanger developed it in the 70s. And using Klinov fragment was not always successful to come up with the good sequencing results. उसकी रीज़न क्या थी? उसकी रीज़न में कुछ ड्रॉबैक्स थे जो कि Klinov fragment की यूटिलाइजेशन से सामने आए. नंबर 1, it was not highly processive. Means we can say कि इसकी efficiency जो है या इसकी जो स्पीड ऑफ एक्शन है दैट वाज नॉट एज क्विक एज वन कैन एक्सपेक्ट ड्यूरिंग द मॉलिक्यूलर बायोलॉजी रिएक्शंस. Secondly, it could not read the areas of DNA which have secondary structure. We can say it could not traverse those areas having the secondary structure. So ultimately, this enzyme left from the point where the secondary structure of the DNA is present or was observed. Thirdly, this enzyme preferred the utilization of dNTPs from the mixture instead of ddNTPs. So, if the enzyme is choosy or selective about using the one type of nucleotide as compared to other type of nucleotide, then sequencing becomes hardly difficult process. During the DNA extraction or DNA isolation, every time we come across with the contaminants. So in the presence of contaminants, if left behind during the processing of the DNA, then this enzyme or its activity was highly effective due to those contaminants, and the results were not produced by the experimental work. It could work efficiently only with the single-stranded DNA, but not with the double-stranded or denatured double-stranded DNA. So the limitation was there that we could only think about sequencing the single-stranded DNAs. So these were those handicapped situations which left the scientist with no other choice, because due to these conditions, barriers, it was difficult to sequence the larger sized fragments of the DNA. Therefore, scientists started looking for other enzymes and they came up with the sequenase or tag DNA polymerase and its variants, which are nowadays successfully being used in the sequencing reactions.